All right, everybody, welcome to our EDS seminar. Today we have a working group in town, and they are here to give us a presentation about the really cool stuff that they're doing as a working group. We have the two leads, Daijiang and Benjamin, down here, and they're going to talk to you about some cool urban invasion biology. Welcome back.
Well, thanks everyone. It's so excited to be here, and particularly in this really nice weather in Boulder. So we're really kind of enjoying our week here so far. So today we're going to talk a little bit about the work we're doing here. So try to understand global urban biodiversity, and particularly focus on species invasions of plants. Okay, I will talk briefly about the work we have done so far related to the data sets side, and then Ben will take it over to talk about some of the research projects we have done and also we're currently doing. Okay, right. So this is a figure showing the global human population growth, and we all probably have seen this before, and we can see over the past several hundred of years, the global human population growth is really beyond exponential, right? And particularly currently, majority of us actually live in urban areas, and this made the cities the most larval habitats for species biodiversity. Particularly, we can see urbanization have different impact on different taxonomic groups, and And on top of that, cities are really good loads of species movement, because you think about all this globalization and connectivity between cities and across them, and when we move things around, chances are we're going to move lots of species around too. So that's going to have lots of species invasions happening. And we think cities, why being so effective as nodes for species invasions, because there's 4 different access of cities. So the first one is the connectivities, right? So when we have the flights and we have different roads or shaping between different cities, we're going to move species around. And so that's one of the important aspects. And another one is the physical properties of the cities, and that's going to have an important impact on whether introduced species will be able to establish and thrive, right? Because of all those like building density, road density, and also green space, and that's going to have an impact on which species will be able to thrive there. And third, cultural and socio-economic history are also important. There's the existing studies already showing that wealthy, maybe historical neighborhoods have higher diversity as well as higher number of exotic species, right? So that's going to have important impact. And of course, biogeographic climate probably still is one of the most important driver of species invasion and biodiversity.
Hey, sorry, real quick.
Yes.
It looks like online you're presenting still the PowerPoint in the slides. It's not showing the full.
Okay. Thanks. Let's see whether we can fix it.
Share, yeah, like
share again, and this one. Is better?
Yes.
Okay.
Sorry to interrupt.
No. Thanks for letting me know. Actually, that's probably important for those on Zoom. So sorry about that.
No worries.
Okay, great. So that's all the 4 aspects we think, why cities would be important to study in terms of species invasion as well as biodiversity. Of course, on top of that, we have policy and management, right? So all this policy maker and also how we manage our cities, green space, parks, all this type of thing is also going to have important impact on species invasion and biodiversity. So ultimately, the question we are really interested in, how does urbanization affect the species invasions and potentially beyond that, like looking at biodiversity in general. And organized by this interesting question, so we put together this global urban biological invasion consortium, and over the past several years we have multiple working group meetings hosted by IDIF, that's a German integrative biodiversity center. And so from there, one of the main goal of those working group meetings is to build a database of global urban species invasions. But of course, if you think about that, then the immediate question is, okay, what is an urban area, right? So here we just use the global human settlement layer. So they provide this database of human settlement layer, like they call urban centers. And so here is just two of them showing Denver here and Boulder here. This is just a zooming of the Boulder, and you can see this polygons is just like the urban center defined by this global human settlement layer. And so they are kind of a threshold is like they're looking at 1 kilometer spatial scale, and then within each grade cell, if you have more than 1,500 human, and also there's a threshold of build-up environment too, so a combination of both, as well as you need to have at least 50,000 human, and this continues gray cell will form as an urban center. So it's more data-driven definition of urban center instead of, let's say, administrative boundary or those type of thing. So that's how they define urban area and or urban center. So across the world, we have more than 11,000 urban centers. And then for each urban center, we went through, try to find, okay, what kind of species we observed within this urban center, right? So for that, we have two types of data source. So the first one is the literature search, and also we have an extended international kind of collaborative network. So we kind of calling for data, and we have collaborators that contribute data, as well as previous network like the Urban Biodiversity Network. We already have built a very impressive data set there. So we're standing on those existing data sets. And also we try to search additional data repository published and then integrate those together. And we also have a second source that's query the GBIF, that's a Global Biodiversity Information Facility. So that's probably one of the most central and important biodiversity data sets. So we just query within that polygon of this urban center, each urban center, okay, what kind of plants we have here, okay. So after some data cleaning and integrate and also do quality control, which I'm not going to go through into the detail here. If you're interested, happy to chat about that later. And then we integrate these two data sources together. And so that gives us a list of plants within each urban center, right? Now, the real fun thing starts. How about the species names? If you have ever dealt with, hundreds of thousands of species, you know how important but also tedious it is to get the species names right. So we just check them with we use a word checklist of vascular plants as our taxonomic backbone to kind of cross validate our species names and integrate them together, standardize them. And then now we have a standardized name of species within each urban center. And then the next part would be decide, okay, who is not native here, right? So we need to compare and contrast with the database. That's a global naturalized flora. So that's a database basically document at where and what kind of non-native species or naturalized species you have, right? So we need to do that. Okay, once we go through the steps, ta-da, we have this database of Cubic. We call the Cubic data. So we have basically essentially a list of plants, including both native and non-native actually. So with 553 urban centers across the world, and here's a map. So that's a basic distribution. You can see obviously there's some spatial gaps, like for example India, we just don't have any city there, and simply just we do not have enough high quality data for these urban centers in India, as well as lots of other countries. So if you have data, if you know somebody have data, can potentially be helpful or useful, feel free to get in touch. Happy to incorporate as much data as possible. And over those data sets, we have 8,140 non-native plants here. And this is the data paper we just published, and it has all the details about the quality control and all those thresholds we used. So yeah, I think that's it for my part, and I want Ben to take it over to talk about some of the projects.
Thanks for the tag team. All right, so what I'm going to talk to you guys about today is some of the questions and so again, this is a big collaborative effort using this database with our group. And so the main aims of this study were to create comprehensive lists of species that are widespread and meaningful for us to better understand how cities were being populated by these non-native species. Secondly, looking at whether or not cities that are on different continents have kind of a similar group of non-native species that are inhabiting them, whether there's some homogenization going on, or whether they have different non-native species present in cities worldwide. And then finally, to better understand the origins and as well as the impact of urban non-native species across our 553 cities in our database. So the GUBIC data, which you guys are now a little bit familiar with based on Daijiang's description. So we started with 7,792 non-native species, and so yes, you do remember a couple minutes ago that number was different. So we removed archaeophytes from the database for this analysis, so that's why we have fewer non-native species to start with. And so we did a series of filtering down these data to what we thought were meaningful lists about widespread species. And so the first list, we have our continental list. And so this is for each individual continent. We looked for species, non-native species that were found in greater than 70% of the cities on that continent. And so in an instance where we didn't have 10 widespread species on a continent, we lowered that threshold and included enough species to get up to that 10 species. And so we can see here for the individual continents, Oceania and North America have the most widespread non-native species. Again, species that are found in greater than 70% of the species that, of the cities on that continent. So again, the continental list here. Then we have a list which we call the omnipresent list, and these are simply species that are found across all 6 continents. We're excluding Antarctica here, and so this is kind of that omnipresent global list of species that we can find in at least one city on all 6 of these continents. Because we have a different sampling, different number of cities on each continent, we wanted to do a little bit more of a conservative list. So for example, some of these species could be, they're just found on one city in each continent. So you wouldn't necessarily call them widespread. So we added another layer of filtering to get our most widespread list. And so you had to be on that omnipresent list. You had to be on all 6 continents, but you also had to be in greater than 70% of cities on at least one continent. So you're not just a species that happen to be found in one city on each, at least in one continent. You're really, really widespread. And so again, this is our most widespread list, 96 species. We did a couple different thresholds to kind of find a meaningful list. When we had really, really high threshold of number of cities, we excluded whole continents. And when we had a really, really low threshold, we had hundreds and hundreds of species that were harder to kind of grapple with. So this was kind of our ultimate decision to get a manageable list of species that were widespread, found on all continents. And so that's how we found, again, so the 4 different kind of lists that we're going to be kind of looking at here is, again, the 7,792 totally non-native plant species across all urban areas, our continental list, our omnipresent list, and our most widespread list. And so let's dig in first to our full list of our non-natives across our cities. And I know this is a little bit of a complicated graph, but a very cool graph that our collaborator Lauren Traudat came up with. And so what we have here is that we have a bar chart where each bar represents the species richness of the intersection of the continents that we're looking at here. And so this first group right here, you can see that the color from the continent is highlighted when we're looking at that. So this bar here corresponds to species that are found only on Europe, so non-native species that are only found in European cities. And so these first 6 bars are for species that are only found in that given continent. And so again, for Europe, there's 1,361. North America is next with 1,132, and so forth for each of our continents. Next, we have the intersection of all pairs of continents. So these are non-native species that are found in cities only in that pair of continents. So Europe and North America have the most up here, 496. Next, we can see that North America and Oceania have 277, and so forth for all pairs of continents. So we did the intersection for 3, 4, and 5 continents, and then when we get to 6, we're obviously at our omnipresent list, those 302 species that are found across all continents. So there's a couple interesting things to look at here. First, we can see that, species that are only non-native in cities on a single continent tend to be have the largest richness or the most common distribution, and that decreases when we go from 2 to 3 to 4 and all the way to 5. But one thing that's kind of interesting here is that our species on our omnipresent list, so our 302 species that are found on all 6 continents, are greater than any combination of 5, 4, 3, and almost all of the pairwise continental comparisons, except for North America and Europe. So we're really finding this kind of group of species that are found on all continents, these urban non-native species that are repeating themselves in cities across the globe. You can also sum across each of the continents to get the total number of non-native species in cities on the given continent. And so we can see North America has the largest number of urban non-native species, whereas South America has the fewest, and you can see in between. And so this is kind of just an overall conceptual view of all of our non-native species and where they're found and what continents they're shared between. So next we're going to dig into our continental non-native, so it's species that are widespread on each continent. And we're going to look at the origin of these species. And so here we have, on this side of our plot, we have the origin, and so the number of species that are on that continental list that are from these donor continents. So Oceania has very few species that it's donating to become urban non-natives in other continents, whereas Africa, temperate Asia, and Europe have larger numbers. Over here, it's just the recipient continents, and each one of these kind of ribbons that goes across is showing the proportion of non-native species in the given continent that's donated from the origin continent over here. And so I know you look at this psychedelic spaghetti here, and you're like, okay, I see some colors. And so maybe your first inclination is to say, okay, everything is going everywhere, and you wouldn't be wrong. There's a lot of kind of little things to pick through here. A couple that I'm going to point out. The first is that temperate Asia is the main donor of species to urban centers on other continents, followed by Europe, and then Africa after that. So a lot of the species are coming from there. And then another interesting thing is that asymmetrical exchange between the Americas. And so North America has donated 15% of species to South America here. So when we look at our North American ribbon going over to South America, but South America has only donated less than 3% to North America. So we have this asymmetrical exchange from 2 continents that are right on top of each other in relatively close proximity, which I think is an interesting facet that this figure shows. There's probably a lot of other things to kind of pick through here, but we're going to move on to our omnipresent list. And so again, our omnipresent list of our 302 species, these are species that are found on all 6 continents, and we looked at them in 2 different types of ordinations. So this ordination is an ordination of the species. So it's a species by continent matrix that goes in, and in each cell of that matrix is the proportion of cities that the non-native species is found in. And so species that are found close together have similar proportions across the continents, whereas species that are found far apart in ordination space have very different frequencies across continents. And so what we can see along this first axis is that over here on this side we have species that are very infrequent, but obviously still found on all 6 continents, and maybe they're found on 1 or 2 cities on each continent. Whereas The taxa that we see over here are found at high frequency, usually at multiple, if not all, continents. And so it's kind of giving that first axis is that kind of gradient of really infrequent to high proportion. And then this second axis kind of splits out over what we kind of conceptualize as kind of a tropical temperate split here. And so species that are up here are found at high frequencies in Asia and in Africa and South America, so continents that have tropical components, whereas species that are found at the lower side of the second axis tend to have high abundances in more temperate continents like North America and Europe, and then Oceania and Asia kind of split the difference. One thing to point out is that species that are found right across this midline right here, over here on the right side, are species that are at high frequency across all continents. So like Erigeron canadensis, which some of you may know, and I should know this from our database, but that might be our most frequent. So that's our most frequent non-native species in cities across the globe. So this middle part, these species here are ones that are found at high proportions across all continents. We also did a kind of a more traditional community ecology site by species ordination or city by species ordination on presence-absence data for our omnipresent list here. And a couple things to look at here is first that Europe is kind of really displaced in ordination space. It's kind of over there by itself on the right side of the ordination, whereas our other continents are all kind of stacked upon each other. So we're seeing that Europe has a different composition of these omnipresent species. Also, both Europe and Asia have those kind of over-dispersed convex holes that we see right there. So they're more variable in their composition of these non-native species. But even though this data cloud for Asian species has this large dispersion, a large chunk of them are kind of found here that are overlapping with those other continents other than Europe. And so the thought process here is that Europe is a big donor of species, so they're donating species to a lot of these other continents in terms of the urban non-natives in their cities. And they're also kind of resistant. There's been so much time that European cities have been farmed and species have become kind of adapted to this disturbance that a lot of the non-natives don't really spread as well across those cities. And so that's why it's kind of that outlier hanging out there while the other continents are kind of stacked upon each other. Next, we're going to get to the impacts that some of these species have, right? We think about non-native species, we worry about them in urban systems and in natural systems as well. And so we took our most widespread list, so there's 96 species that are found on all continents and are also widespread on at least one continent. And we looked at their ICAT scores to see how the impact they may have on the cities that they're inhabiting. And so we can look at these different classifications from a massive impact, to major, to moderate, all the way down to minimal. And we can see that 15% of them have a moderate impact. And we can see this kind of little distribution going on here. But obviously the elephant in the room is that 2/3 of them are not assessed. And so these species that we identified as being everywhere and being widespread, we don't know that much about. We can look at a couple of those species. Black walnut, which has massive impact, and then a couple other species that you might be familiar with if you study invasive species or hang around in cities. But again, really, this is kind of looms large, the fact that 2/3 of these species that we identified, we don't know a lot about their impact. So what can we conclude overall? So human activities have created this kind of well-defined urban florome, which is kind of this fingerprint, this repeatable pattern of common species that we're finding across our 6 continents. But we really need to know more about those widespread species. We saw that we have a huge dearth of information on their impacts, and that's something that it could be really important in thinking about managing cities and understanding the impact of invasives and non-native species. And of course, this is a dynamic situation going on here, right? There's new species that are being brought into cities as we speak right now, establishing, spreading. And so updating this widespread list, keeping current tabs on it, as well as, again, looking at those impacts is going to be important for understanding invasion dynamics. And so this little study, this little exercise gives an idea of where these non-native urban species are in cities across the globe. But species are a lot more than their names, right? They bring their traits with them, they bring functions. We're also interested in understanding a little bit more about those species traits, about those functions. And that's what we looked at in our next study. Oh, and also, sorry, if you're interested in more of this, some of the decisions we made, a lot more, you can find that here. But on to understanding traits and functions of these urban species. And so as Daijiang had talked about, we know that cities are highly modified habitats, maybe even novel habitats, although I've been told I shouldn't call them novel. And so we can think about the different habitats and the different conditions that non-native species face as they assemble into a community, whether it's impervious surfaces, habitat heterogeneity, eutrophication, heat island effect, fragmentation, and just really modified systems. And so the question is, how are these species assembling there? How are they coexisting? What kind of communities are there? And that's a question that we're really interested in understanding and thinking that traits might give us some insight into how those species get there and how those species coexist. And a way that helps me kind of think about how species get from this global pool of species all the way down to a sewer grate in the city is this urban filters framework that was developed by Mila Aronson, who is part of our working group and sitting right here. And so I like to break this kind of filtering framework down into two parts. So the first part is thinking about how do species get into the urban species pool for a given city. And so one route is just from the regional pool. They filter down from the regional pool. And how does a species get into a regional pool? Well, this is classic ecology. So we can think about biogeography, evolutionary history, climate is going to result in this regional species pool. And then some of those species filter down into the city pool. But we can bypass this regional species pool with this human facilitation of non-native species. Humans are bringing certain species right into cities, kind of bypassing that need to get into that regional pool. And so this is going to give us our species that are there in our regional pool. Once they're there, they have to go through a series of filters to kind of land in a specific habitat within a city, like a sewer grate. And that's going to have a lot to do with kind of the urban forms, so the structure of cities, the history of cities, socioeconomic and cultural aspects of cities. And then some more classic, ecology, such as species interactions, are there pollinators, herbivores, competition with other plants, and then human facilitation as well. People still move species around within cities as well, and that's going to let us know who is going to be in our given habitat, who's going to be in the lawn or in the sidewalk cracks or wherever it may be within a city. And the cool thing about this is that traits are really mediating which species are making it through these set of filters, and so they're kind of a really useful currency to think about how species kind of assemble into urban habitats. And so the question that we're asking here is, do urban environments filter for species with similar traits regardless of provenance? So does it not matter if you're native or not native? Is it such a harsh habitat that you have to have these traits in order to be there, and there's no difference between native and non-natives, or are there differences in how those different species groups are assembling into cities? And so we use trait space, the global spectrum of plant structure and function, which is a global urban trait space that was developed by Diaz and colleagues using these 6 plant traits over here. And so within this framework, it's conceptualized as the first axis of a PCA on those traits being structure. And so species that are found with positive PC1 values are taller species that have larger seeds and more dense stems. These are often woody species. That's this kind of concentration over here are woody species. And then species that have negative values on PC1 tend to be shorter, smaller seeds, less dense stems. And so these are more often our herbaceous species, which you can see in this cluster over here. This second axis is the leaf economic spectrum, or leaf function. And so species that are found with large positive values up here tend to have acquisitive leaves that have high SLA, high nitrogen, large leaf area. Whereas species that are found with negative values on that second PCA axis tend to be more conservative, tend to have greater leaf mass area, lower nitrogen. And so this plant trait space is a really useful framework, useful tool to think about species and where urban, native, and non-native species might fall. And so that's exactly what we wanted to do. And so voila, we have Gubic, which is an amazing database, which you're learning about today. And so again, our 553 cities across our 6 continents, we're able to use 2,777 of those species. I know you're looking at that and saying, but what about the other 5,000? Well, in order to put them into this trait space, we need the traits. And so we need these 6 traits. And so through different trait databases, we were able to get these 6 traits for 2,777 of them, a relatively even split between natives and non-natives. So you're native if you're not found as exotic in any cities in our database, and you are non-native if you are found as exotic in at least one city in our database. And so that's how that division is made. And so we put these into a PCA, into that global trait space to see if native and non-native species were found in the same place or different. And so this is what our trait space looks like. We have our gator blue and orange here. Orange is for non-native, blue is for native. And the first thing we can see overall is that we recovered a similar trait space to Diaz et al., which is good, meaning that it is maybe truly a global trait space that kind of these axes kind of really orient where these species are in terms of their function. And so we can see that again on the positive side of PC1, it's that structure axis. So we have woody species that tend to be taller, larger seeds, denser stems. And on this negative side, we have this smaller herbaceous species, smaller seeds, less dense stems. And then we have our second axis again, where leaf nitrogen, leaf area, the inverse of leaf mass area is SLA, which would be up there. So we have those acquisitive leaves, and then we have those conservative leaves down here. And really, we see that a lot of our orange is on this side of the PC1 structure axis. So we have a lot of herbaceous, non-natives, and then we can see over here that we have a lot of woody natives over here. And we can compare the distributions of those PC scores, which is what we did here. And so when we look at PC axis 1, and we can see that, yes, absolutely, we have a high concentration of native species that are woody, that have these larger structural attributes. And we have a lot of non-native species that have the smaller structural attributes on here when we look at that structure axis. For this second axis, this leaf function axis, if you turn your head and close your left eye and squint your right eye, you can see that maybe the orange is popping out a little bit there and the blue is popping out a little bit there. And so it appears that non-native species are a little more acquisitive, but the significance is largely due to sample size there, so that's much less strong of a pattern that we have there. So what can we conclude based on this ordination space when we consider all the taxa together is that our non-native urban species are shorter herbaceous plants, they have small seeds, lower stem density, and more acquisitive leaves, while our native urban species are taller, woody species, tend to have larger seeds, greater stem density, and less acquisitive leaves. We also took a more holistic approach to kind of looking at the trait space in multivariate trait space using hypervolumes. And so we built null distributions for what we'd expect in terms of hypervolume overlap between native and non-native species using bootstrapping. And we did that for all species together, just herbaceous species and woody species. And we compared that null distribution to our observed overlap in multivariate space of the hypervolumes. And that's what that red line is. And we can see here for all species, herbaceous and woody species, that the observed overlap is a lot less than we'd expect under the null. So we're seeing that again in this hypervolume space, that native and non-native urban species have a difference in their trait space. Because herbaceous species and woody species have such different life histories, we see that's a lot of what's driving our pattern when we have all these different taxonomic groups together. We also decided to do an analysis where we broke them apart to look at herbaceous species on their own and woody species on their own using phylogenetic generalized linear mixed models. And so a model each for herbaceous and woody. Again, our response variables were our PC1 and PC2, so our structure axis and our leaf economic spectrum. Our predictors were provenance, so native or non-native, temperature, precipitation, and interactions between provenance, and our climate factors. and then we use a phylogenetic tree to control for evolutionary history. And so I'm going to be showing you these model predictions from each of these models. So this is showing just for herbaceous species using PC1, so that plant structural axis, as the response. Again, if it's a positive value up there, it means it has large structural attributes. Negative values mean it has small structural attributes. We can see the predictor variable is going to be here. We have a rug showing where the observed values for temperature are. And then again, orange is going to be non-native, blue is going to be native. And so what we can see here with temperature is that at low temperatures, there's not much of a difference between native and non-native in their structural component. They both are relatively small. But as temperature increases, there's a significant interaction where native species become larger on that structural axis, whereas non-native species decline. And so we can see that difference there, that interaction and difference between native and non-native across that temperature gradient. When it comes to precipitation, there's no difference between native and non-native species, but we can see that they both increase in plant structural size for that first axis with precipitation. Moving on to woody species, again, there's no difference between native and non-native woody species on that first PC axis for structure, but they both increase with temperature significantly, and there's a slight decrease with precipitation in that plant structure axis for both native and non-native with precipitation. So when we get into the leaf economic spectrum, so that PC2, we start to see some differences here. So the non-native species tend to have more acquisitive leaves than native species across the temperature gradient. They both decline, but we can see that difference is maintained. When we look at precipitation, we see an interesting pattern here where in really dry conditions, both native and non-native species are having conservative leaves. But as precipitation increases, they become more acquisitive, but the non-native species do so at a greater rate. And this is a marginally significant interaction, so p-value of, I think, 0.1 for those of you counting. But we see there's a significant difference in native and non-native there. And when we get to the woody taxa here, we can see that in cold conditions, that non-native taxa tend to have more acquisitive leaves than their native counterparts. But as temperature increases, that difference goes away. And so we have a significant interaction for that. And when it comes to woody species along that PC2 leaf economic axis and precipitation, we see the nearly identical pattern that we saw up here for herbaceous species, where in those really dry conditions, native and non-native species are both similarly constrained to having those conservative leaves. But as precipitation increases, both native and non-native species become more acquisitive, but the non-natives do so at a greater rate. So how does this fit back into our urban filters framework? So when we think about our first chunk over here, what we found out is that herbaceous species are dominating non-native introductions. And this is true outside of cities as well. We know that herbaceous species tend to be a larger proportion of exotic species. And it might be that those smaller seeds help facilitate transport into the urban species pool. And so those attributes of those urban species along that first PC axis facilitate kind of this pathway into that urban species pool. When it comes down to our next filtering, to how does a species get to its given spot, its given habitat within a city, these non-native species, maybe they're getting filtered into these kind of highly disturbed habitats like sidewalk cracks or sewer grates or right-of-ways that require them to have this rapid growth strategy, less structural investment in their leaves, this kind of live fast, die young strategy to really kind of just make it in that disturbed habitat and really have an adaptation to highly disturbed habitats when they're kind of filtering down into their specific locations into a city. On the other hand, the natives might not have those same constraints. So they have greater structural investment in their leaves. longer lifespan. So it's possible that these native species are in remnant habitats that are not as disturbed, parks that allow them to have more woody species, have longer lives, and have a different strategy than those non-native species that are getting filtered into those potentially highly disturbed areas. And so what can we conclude overall? So urban native and non-native species differ in trait space. We can answer that question. We looked at it a bunch of different ways, together, in 2 dimensions, in multiple dimensions. Non-native species have more acquisitive strategies, so they have less structural investment in their leaves. Natives have more structural investment in their leaves. There are some interactions with climate, so there might be kind of climatic filters that make native and non-native species have similar traits and be in a similar trait space, like we saw with under dry conditions. But once that filter is released, we're seeing that a lot of these non-native species are having more acquisitive leaves, and so that interaction is something that to be explored further. And so I kind of perceive it as different filters. There's different filters to get into to get into the urban pool. The native species are already there. They're already remnant. They don't have to get there, whereas the non-natives have to be transported there. And so those herbaceous species, smaller seeds, might facilitate passage through that filter. And then when we get to our local spot, within the cities, again, they might be subject to different filters there, whereas and can have more conservative leaves and have a little bit of a different life history strategy. And so what's next? We're here at ESOL, which we're very excited about. And we're really interested in better characterizing cities. So I'm an ecologist, and I think about a city, and I'm like, it's big, it has a lot of people, it has a big area, or can those be my covariates? And we know there's a lot more information, socioeconomic information, historical information, that could bear upon our understanding of native and non-native species in cities. And so that's one of our big aims for our group here. We want to get more trait data. So is my hand waving true? If we go out here and we see, are we finding these acquisitive species in sidewalk cracks in disturbed areas, and are we finding these greater structural investment in these nice little parks? To test that, we need local traits, and we can know what's going on in cities, and so that's something to move forward to. And then to expand to other taxa. I love birds. I'm obsessed with birds. And so think about other taxa in terms of native and non-native species and their traits, and just their distributions across the cities worldwide is something that we're interested in doing. And that's all we have for you. Thank you for coming out and being here for the talk. I'm happy to answer questions now, but we're going to be around all week, so come by and say hi to us and ask questions about invasive species. And of course, all of the funding sources and that'll allow us to get together and discuss these ideas. And so thanks again. Yes.
I think just a definition question. You're omnipresent.
Yes.
How can something be invasive everywhere? Doesn't it have to be native somewhere?
Right. So in some cities on a continent,
okay,
it's outside of their native range. Okay. So yes, it's a very good question. Makes total sense. But yeah, so there are some that if it was only native in a given continent, then it wouldn't be included on that. So there has to be cities that are outside of its range. Yes.
Curious how much these traits are actually helping these non-native species dominate urban areas versus maybe it's like cultural trait that people are preferring some of these species, and maybe like when you see North America and Europe dominate these counts in Oceania, is it like maybe there's some way to look at like a colonial history of people trying to like impose certain species in these areas? That's kind of what I got from that.
Yeah, I mean, so that's a great point. And so thinking about the colonial history, clearly Europe as kind of a donor, has some, is kind of evident of that pattern. And so I'm sure I'm sure there's been papers that show that people are selecting for species with specific traits, whether they're bright colored flowers, whether they're nice leaves, whatever it might be. And so there's no ornamentals that are included in this, so that kind of takes that piece out. Yeah, I mean, I think the key thing here is that humans are in cities, and we want to better integrate humans and their cultures and their choices into this. And so that's part of getting better covariates to understand. And so, yeah, it's an interesting thought to think about that relationship between these, non-ornamentals, but traits that they have, and whether it's just, it's a sidewalk crack, or if there's another component of the species or of humans that are specifically selecting for that. So, yeah, it's a really good question.
So most of the widespread species are accidental, right? And so we look at that PCA with all those species names on the end. None of those are ones that people would purposely introduce. So that's why they're coming out in the trait space, is because they have these characteristics that allow them just to hitch rides and take advantage of openings.
Interesting.
Thank you, Mark. Yes.
Thank you for your great presentation. And I wonder how this project have treated or we plan to treat essential biases. For instance, the existing global biodiversity data sets are biased toward North American and European countries, and largely missed in the South America and Africa continents. And also, your urban maps show that there are large, huge missing across the tropical regions, which are biodiversity hotspots. Yeah, and I wonder about that.
Yeah. So yeah, you can look at our map. It definitely has some gaps, and we thought, and we made an effort to do well here. And so yeah, so in terms of biases, we looked at things like rarefaction curves for the species that we're finding for each city, and kind of figured out what we thought were kind of robust samples for the cities, and so that's why some cities got excluded. But yeah, we need more data on species, on traits, and as you're talking about tropical areas, that's where some of the urbanization is expanding most rapidly, and so it's a very good question. And so one of the cool things about this group is that when we had our first meeting in Toronto, we had, I think, people from over 40 countries, and almost, I think, all continents. And so we're trying to build this network where we have folks, we can get data from GBIF, but we also have colleagues who are doing urban ecology in cities worldwide. And so yeah, it's a challenge, try to build out to the gaps on this map and get robust estimates of non-native species or native species for cities is something that's a challenge. And so, I mean, Daijiang might be able to talk more. Other people in the group who work in some of these regions who that are less represented, and yeah, it's a challenge and one that there's not that great answer to, but we're I think that expanding that network and not just going outside and collecting plant traits or surveys from our northern hemisphere universities and expanding that to other areas is going to be something that's important to get the full picture for sure. Yes.
Have you happened to study that the vectors of spread of these non-native species, for example, like who might bring them, like human migration versus like can we trace back to the bird diversity have changed, like if the climate or something, who might happen to have carry these species?
Yeah, so I mean that's a great question, and that's kind of part of what we're putting together now is this set of covariates, predictor variables, whether it's shipping, whether it's flight, whether it's colonial history, whether it's just geographic distance, and so yeah, those are kind of the next questions, and then a key part to understand this global picture is to get the information on city attributes and their relationship with each other to better understand that process of spread. For sure. So yeah, that you're right on point to ask that question. Yes.
I was curious about the ICAT impact assessments.
Yes.
Because obviously impact's pretty context specific.
Yeah.
And for the 90 or 95 species that are everywhere, did you find that the assessment did a good job of characterizing local impact and then relating that across the entire as an impact score for the entire species? And then do you think that because those species are everywhere, assessing their impact is difficult via that mechanism, and that's why there were so many species that were left off that impact?
So I didn't do the ICAT analysis, but from looking at this, my understanding is that the ones that are assessed are ones that are known to be hugely problematic. And so I don't know if we can say that the ones that aren't assessed yet aren't like driving land managers crazy yet, or, you know, so it's not like, oh, there's this terrible species that's taking over everywhere, but we're not. So at least that's my, you know, understanding of how we got here. But you know, I don't know, Mark, if you have some more insight into ICAT and why there's this kind of gross distribution that we see here.
There are a bunch of species that are unassessed that we know have large impacts.
Okay.
But usually it's a smaller number of cities. So there are some major data gaps. But the one thing that's interesting is like some of the photos you're showing with my name underneath them
Yeah.
were those big impact species, right? And so the one growing out of the wall, the one coming out of sewer grate, those are the ones that have the high impacts. And so if we see them, they're more obvious. And they tend to be woody ones that we do the assessments on. There's a lot of herbaceous ones that have, especially ones that release chemicals and allergens and stuff that we haven't assessed in the same way, but they do have impacts.
Yes.
Sorry, I have not read your paper. How do you distinguish between invasive species, non-native, and exotic species?
Well, I don't.
Huh?
I don't, and we didn't. So in this paper, it's just non-native. This ICAT is kind of an assessment of, I could push you towards saying that this species is invasive because its effect, because of its impact. But so far, in our analyses, we haven't, we haven't circled in on a firm designation. Otherwise, it would be obviously very interesting to look at the trait space of the invasives, the non-natives, and the natives, and see if they're different. So yes, so we we don't have a specific designation as invasive yet, but I mean, hopefully we'll get to that. But yeah, it's.
So when would you think that non-native, by definition, non-native species are exotic species?
Yes.
Yes.
Are they all invasive?
No.
So
Did I get it right?
I think such research has important implications for human society. That's why I'm not sure.
Yep.
But it seems like it has an implication to many readers. Non-native species are bad, or at least ecologically bad, but I don't believe that.
Yeah,
What
I
about
mean,
you?
Not
More than 85% of stuff we eat, we drink, actually come from exotic species, non-native species.
yeah, I mean, I think for me, I'm just, interested in, is there something fundamentally different about them?
Yeah, one is legal, one is illegal.
Yeah.
There's fundamental difference between exotic species and invasive species.
Yeah, I mean, I don't think we can go out, or should we go out and manage and get rid of all non-native species. Obviously, that's not even possible. But from an ecological standpoint, is it just run-of-the-mill community assembly that's just happening at over time, or is there something fundamentally different about the ecological processes of community assembly with non-native species, with invasive species? That's from a theoretical interest. That's where I come in. Is it just, hey, if you looked at community assembly of plants for 2,000 years, yeah, things come later, and there's pulses, and there's not. And so, is there something fundamentally different ecologically? Not thinking about the impact, or is a question that kind of fascinates me personally. But I'm sure they're tying that to management and tying that to resources spent and to manage non-native species, or the ones that are invasive that do have huge impacts is important to do. Any other questions? All right, well, thank you very much.